All right, so let's get going. Using INAT for mycological data. So we're going to go start with just what is iNaturalist. If you've never heard of it, all good. Where does INAT go is kind of where I want to focus. So you may be familiar with posting a picture on INAT, but not really know where that goes or what it does for the community. And I think that'll last about half an hour, depending on how many questions we have. And then for the second part, we'll talk about how to actually do it. So if you have any pictures that you want to upload, or if you want to practice identifying as part of the community, we can do that together. You can ask questions. Anyone can share their screen, and then we can definitely do it together. Karen, you said screen is tiny. Is it good now? For me, it looks full screen. You're just muted, Karen. I can't hear you.
I cannot see your shared screen. I see a skinny little line that says INAT presentation saved to my Mac.
All right. Well, is that true for everybody? Maybe I'll just stop sharing and open it again to make sure that it's
I also only saw a tiny little line.
okay. Well, we don't want a tiny little line. Okay, is that better? Awesome. Same window I clicked, so I don't know what's going on there, but that's all good. Yeah, so that's just what I was saying here. So part one's going to be the basics, and part two is going to be us doing it together. So first, this is what iNaturalist looks like if you open it on the web. So from your computer, you'll get this big screen. And if you click Explore, you get all these random observations that anyone has uploaded. You can see this is like when I was making this is what I opened. I'm on iNaturalist.ca, so it automatically chooses everything in Canada for me. That's really handy. You can go to that location tab on the top if you want to make it Alberta, or if you want to go broader to the world. You can totally do that. And right now it has every observation in Canada, so it's almost 13 million. But you can go into the species tab and you can type anything that you're looking for. If you wanted to look specifically for fungi, if you had something really specific in mind, you could just type that in there, and it'll only pull up the observations that are tagged with that name. So super handy if, say, you wanted to go look for morel mushrooms and you wanted to know where they'd been seen in the past, you can use that search tab and search for exactly what you're looking for. And then this is also, so same thing, I did exactly that. Where can I find morels in Canada? And you can look at it as a grid or a list, but even better is when you look at it as a map, because then it'll pull up exactly the data point of all of those observations. So you can see here, I searched true morels. I got 3,000 observations in Canada, and there's kind of like a little mini list on the side, as well as all of those data points. So if I clicked any of those data points, and I'll show you at the end kind of what that looks like live, it would pull up that actual observation where the data point was. So super handy if you want to find something close to you, or in any kind of geographical area where you have an interest. So all of these points come from somebody taking the time to upload their picture, which is great. It's really fun to do, and it adds to that big map of data that everyone gets to access for free online. So this is just a little screenshot of if I was adding a photo from the website. I take my picture, it wants me to put in a species name, and then from the picture, it automatically pulls the date, time, and location. So you don't have to note that down. It's automatically part of your photo. It already records that for you. And then when you click on species name, it pulls up a list of kind of suggestions that it has, where an algorithm takes your picture, runs it against millions of other pictures, and suggests the ones that are the most similar that it thinks match. So I have this Polyozellus mushroom here. It suggests a couple options. One of them is Polyozellus, so it's pretty good. But my recommendation is always to take this with a grain of salt. It's a starting point, not necessarily a right answer. So a little bit about this algorithm that INAT uses. It takes a known set of photos that it's trained to identify. So the people who kind of run INAT, the computer scientists, They upload a whole bunch of pictures where they have a real person say, yes, this is, a squirrel. And it teaches it through that. And then the algorithm gets smarter by matching what it knows to what it sees and kind of gets feedback based on what people tell it. So the INAT algorithm is updated very often. They're constantly running new training sets. It's super innovative, really cool, but it's really not good at fungi. Mycology is not its strong suit. So you can see here, I took a picture of a snowy artist bracket, a conch. It suggests perhaps a beaver, a bracket fungus, that's good, a wild rose, a little tree gull, or even at the bottom, you can see snowshoe hare. So while it's really strong at identifying something obvious, like say a moose or a bird, when it comes to fungi, it's not really well trained. And there's also a lot of options that look similar. You can really find its flaws with the fungi for sure. So here's just a larger picture of what was there. So you can see that, I mean, it's clearly not a snowshoe hare, but it tried its best. It saw something snowy and it tried to work with what it had. So just a little bit about how well the algorithm works. A bright yellow butterfly on a plain background, super easy. It will almost always get that right for you. Something that's really hard is, say, a lichen or an organism on another one. So this is a lichen growing in moss beside grass. It gets really confused. It's seeing a lot of things and it doesn't know what it should really assign to that organism. Something like this lichen on a tree is a little bit better because it has kind of a flat, solid background. And so it's easier for the algorithm to find the thing you actually want it to. But again, it's lichen, fungus, it's not really its strong suit. So it might give you a list of suggestions, and you just have to be a little mindful about how you go through and select which option you're looking for if you don't have an idea of what it already is that you're looking at. Does anyone have any questions about this algorithm before I move on? Awesome. If you do, feel free to interject at any time. So I just wanted to bring this up here too. So I put in a picture of a morel mushroom and you can see that I got a lot of kind of similar suggestions. Some of them are for true morels, some are for false morels, some are for verpas, and then you even have this craterellus at the bottom that's quite different. It's bright yellow, it's differently formed completely. But in this list, there's a wide variety of what we would call edible mushrooms. So if you're using iNaturalist to help you ID something that you're looking to eat, I shouldn't have to say it, but I'm going to say it. Don't trust this computer program to tell you what is healthy, what is safe, what is toxic. It's just a computer. It's just running simulations. It doesn't really know. So just use your best judgment, really. It's a great starting point to take to a guidebook and say, hey, is this a Gyromitra? Like, does that match? And you can use that as a great starting point to not have to go through every key in your guidebook and just skip to one that you think might be relevant. Okay, and the great thing about iNaturalist is that it's not just this computer program. So something like Google Lens or Seek or other kind of algorithm-based apps, they don't have a human component. So you're relying entirely on this app. It's just running pictures and hoping for the best. iNaturalist has a human aspect of verification. So if I propose an ID and someone else thinks that it's wrong, or maybe I propose a genus and they can go to species, so be more specific, then it gets updated based on that human interaction. So you can see in this example, I found a lichen. It was weird. I was kind of familiar with what I thought it should look like, but something was different about it. So I just put it at the genus level. I said, I don't really know what's going on here. And I made sure to comment so that somebody who came to my observation could have that context. And Diane came on and she said, well, this is actually two species. That's why you're confused, because the characteristics of one are also on the other. So you couldn't key it because it had both. And after that, she said there's two present. She picked the one that was the most dominant to put the ID on. She put the comment so that it was clear for anyone in the future. And that ID was refined to something more specific. So you can see that I initially just put Peltidra and she made it Peltidra praetextata, and that was automatically updated. So the next person that clicks on this will see the full species name, and they won't just see my initial suggestion. So as you go, as more and more people kind of comment or add to it, the observation is meant to get more and more refined and more accurate. And sometimes it happens that someone uploads something. It happens with these pelt lichens a lot because they look kind of leafy. So someone will say, this is a plant. The next person comes and says, no, it's a lichen. And you'll see that the name, the kind of assignment jumps all around plant, fungus, whatever. But after 2 or 3 or 4 people go through, it ends up being quite refined and almost always correct. And when that happens, we get this research grade assignment. That's not to say it can't be wrong. Sometimes 2 or 3 or 4 people are all just agreeing for the sake of agreeing, and it's not necessarily right, but most of the time it is. And so this was a really interesting story that I wanted to tell because it kind of showed why INAT can make a difference in science as a whole. So here I saw a lichen. And I was like, I don't know what this is. It looks like a yellow lichen that I know, but it's not the same. Something's wrong about it. So I just suggested an ID of the lichen that I thought it might be at the genus level. Keep it general. Hopefully someone comes and helps. Toby came on. He's the lichen prof at U of A. He says, well, crazy. This looks to be the first record of this lichen in Alberta. Can you collect a specimen and send it to me and I'll sequence it? And I was like, cool, awesome. I get to be part of science. So I took it to him, he sequenced it, and it turned out that his identification was correct. So after he told me that, I changed my identification because I trust his sequencer. Someone else came on, they also agreed with the same identification. And then once that ID and sequence was published in a real academic paper, I linked that at the bottom to say, yeah, it was confirmed as this organism. And that was really cool for me because I was visiting my parents' house at this time and I was just like, Mom, what's growing on your tree? That's pretty weird. And it ended up progressing to this actual full entry in a scientific paper about what lichens are in Alberta. And it was crazy for me. And I was an undergrad at that point. It was so exciting. I was really feeling like I was making a difference. And that's really what got me caught on INAT was that all I did was just post the silly lichen picture. But it went through all the hoops and it mattered in the end. It was good. So yeah, this is just where it ended up in that paper. Super cool. I got my name in it. I can't complain. And it's not just me, like it wasn't just this one-time thing. It happens a fair bit where someone posts a picture, an expert in the field finds it and is like, whoa, you've got something there. I read an article very recently about a guy who was visiting New Zealand. He had a candle burning on his balcony or a light, and a moth came to it. He took a picture of the moth, and it was the first recording of this particular New Zealand moth in over 100 years. It was assumed to be extinct. This guy just happened to take a picture of it, and it went viral. It was crazy. Every kind of entomologist was going nuts that he happened to find this moth, and they didn't. That was the other thing. They were like, I searched so hard, and this random tourist got to see this moth. And I mean, he had no idea the difference he was making when he posted that picture. So it's really interesting how just by contributing to the database, even the littlest observation can actually make a big change. So let's go from there. What actually makes a good observation? We've all taken some crappy pictures where we look back at it and we're like, what was I even taking a picture of? And honestly, those don't go very far on INAT because even if you know what you're talking about, maybe someone else can't get that from the photo. But I mean, you could take a good photo, first of all, but what you can also do is add a small description to help someone else know what you're talking about. So if you're taking a picture of like a mossy field, maybe there's a moss and a lichen, specifying which one you want to put as your ID can be really helpful. Just, I want to ID this lichen. For mushrooms, it's really easy to do a simple underside photo of the gills or the pores, super helpful in making a good ID. Same thing if you cut and check for bruising. These are all kind of the same steps that you would take in the field, But if you can make a note of it or take a photo of it, it helps that people down the line can also see that to make your observation research grade. For lichens, taking a picture of any reproductive structures, super helpful. And for all fungi, a substrate description. Lots of times people take home their mushrooms and it's just a picture of it on their table. And you're like, was this growing under a tree? Was it growing on a rock, in moss? It does matter, especially when you're stuck between two very similar types. Sometimes it can be the deal breaker. And the other thing I want to mention is that being overconfident in your ID is usually kind of more harmful than helpful. So let's say I'm a researcher looking for a very specific species of milkcap, of Lactarius. And let's say I'm looking for the downy milkcap. Someone has found one, but they accidentally tagged it as the wrong species. If I search for this species, I'll never find it because it's tagged as a different species. But if I wasn't sure and I just left it at the genus level, so I just put it as Lactarius, I would probably find it because I'd be searching through Lactarius to see if any of those could be tagged as my species of interest. So I find that if I'm not sure, I almost always just leave it at the genus level and I add a little note that says, I think it might be this, but I'm not sure. There's been some research on lichens and how often they're wrong on INAT. And lichens can be a lot harder because a lot of them need chemical tests or microscopy. So again, when I find a lichen, unless I'm really familiar with it, I almost always leave it at genus level. Okay. Yeah, this is just another example. So I found this lichen, I put what kind of tree it was on. Just a little detail that makes its ID a lot easier. And then, so we talk about making a good observation and hoping that people come to identify it and help it get to research grade. That's all fine and dandy, but somebody has to do that. And a lot of people post to INAT and they get kind of frustrated. They're like, no one ever looks at my stuff. No one ever gets my stuff to research grade, but they're also not looking at anyone else's stuff. So if we're all posting and no one's identifying, we kind of get stuck in this loop of lots of data with no confirmation. And so it really, really helps if you help to identify. And some people start groups, they're called Identifridays, and everyone on Friday goes and they help identify things. And it's really easy. So I'll show you how to do it afterwards, but this is just a screenshot of it. If you click Identify instead of any of the other tabs, it pulls up only observations that haven't been looked at by someone else. So if no one has commented on them and they're very lonely up in the universe, you can find exactly those ones in this Identify tab. So here I said I want to identify, and then I made it a little more specific. So I put fungi in Alberta in those search boxes, and it pulls up only fungi in Alberta that haven't been looked at by anyone before. So, I mean, I can go through here like this. You can see that the ones that are at the very broad fungus level, they're just sitting there. You can't click agree. You just have to click on it and provide a suggestion because right now there's nothing really suggested. It's very broad. And same with the genus level or the family level. There's no agree button. You need to go suggest something to make it more exact. If someone has already suggested a species name, so here, like the star rosette lichen, it says agree underneath it, you can just click that button and it's automatically agreed with. I find that that's really helpful if you have something big and obvious, like a beaver or a moose or a cardinal or something like that. And I don't think it's really a good feature for a mycology kind of perspective, because in most cases, we need more than one picture, like maybe the top view and the under view of a mushroom. So I don't like that agree button unless it's something super obvious, like maybe black knot fungus. So, I mean, that's up to you, but I always kind of trend to the side of being more cautious on my IDs than more kind of liberal, just for the sake of not making incorrect contributions. Yeah. So this is just another view of it. So I think I just scrolled down the page to find some more that I could agree with or disagree with or whatever. And you can see that agree tab only comes up for the species. And then at the bottom here on your profile, if you start an INAT profile, it actually keeps track. So this person, I don't think it's mine because I definitely haven't made this many IDs, but they have 400 observations. So those are photos that they uploaded versus 2,400 identifications, which is on this ID tab here. So this person has a really great ratio. They're IDing more than they're submitting. They're actually like contributing more to the community than they're just getting back in IDs. So that's great. Okay, so I'm going to switch my screen here. And I'm going to pull up my INAT. All right. Can you guys see my mouse moving around? Maybe just like a thumbs up if you can. Whee! Okay, awesome. So this is live. So this is actually on my browser. And I just click this Explore tab. It reloads every observation in Canada right now. So you can see it went up from last when I made my presentation. We're now at 13,200,000. So it's going up. And like I said, you can view this as a grid or as a list or on the map. Crazy. And this isn't really helpful to me because it's every observation in the world. So let's instead change the location to Alberta. And let's look for Gyromitra because I think they're going to come up pretty soon. So we can just click that. We can see that in Alberta, there were 161 observations of 6 different Gyromitra species. And we can see our list here. I don't know what this is. Yes, that's what I want. Okay, there we go. So it has this list. As we scroll, it kind of pops up in different places on the map. Or I could click on something specific on the map and that pops up. And I mean, let's say this was a really cool observation. I can actually click on it and it brings me up to this person's observation that they made way up in northern Alberta. You can see that no one has helped them. And that happens a lot. The further you are from a city or kind of somewhere where lots of people live, they don't usually stretch their IDs out too far out of their geographical area. That's just kind of, it is what it is. So you can see they posted 2 pictures, very cute, adorable, love it. So we can also go to your observations. So that's everything that you posted. So welcome to my world. It's gonna load nice and slowly because I have quite a few observations. Or maybe it's not gonna load at all. All right. All right, the identify tab works, so let's go there. So what we can do is this, again, is every observation that hasn't been fact-checked by someone. So if someone uploaded it and nobody helped them, that's where it's living. And so let's look at this one right here, this unknown little flower. So when you tag something only as unknown, it will not come up for anyone who's looking for something specific. So let's say I'm really good at lichens. I can search common lichens up here. And so what will come up is everything tagged common lichens or more specific. So if it's a species of lichen, a genus, a family, whatever, as long as it's tagged as a lichen, it'll come up in my search. However, if we leave it as an unknown, it will not come up in my search, even if technically it is a lichen. So that's why it's really important to choose a broad ID, even if you don't really know what it is. Choosing something even as broad as like fungus or bird really helps someone find it later to help you get that ID. So we can see here, these are all the lichens that no one has looked at or reviewed. I think we're in all of Canada. Yep, so there's going to be quite a few. And if I can agree with it just on the species level from looking at it, I can click this agree tab, but I'm not going to because I don't think it's a good idea for mycology in general. But you find some cool stuff in here. Usually on Fridays, I try to go through and make sure that I pick some that I can identify to kind of make my contribution. Yeah, so those are the main things. We can also go to this upload tab up in the top corner, and we can choose like a photo to upload. I always have something on here. So here's a lichen that we found out in a field somewhere. And you can see that it automatically knows the date and time. And it's trying to pull the location, but someone sent it to me, so I'm not sure it's gonna work that well. But when we go for this species name, that's where that algorithm I was talking about pops up. So here, you can see in the picture, it's not very good. It's a stick with multiple lichens and it has grass in the background, so it's not going to like it very much. But it's providing, I'm going to say like 8 to 10 suggestions, and they're all lichens. So it does know something. I'm not saying it's not smart. I'm saying that's not always right. So it provides these 8 lichens for us that it thinks it might be. And so I can pick one of these, or I don't have to. I can write like anything I want. I can say that it's a moose and I can upload it as that if I wanted to. And then someone would come and correct me later because that's obviously not what it is. Yeah, so it's like I said, everything here is really simple and it kind of relies on one person contributing and another contributing with an ID. A photo and ID takes kind of a community to make it all work, which is, I think, really fun and satisfying. I'm going to abandon this because I've already posted it. There we go. And here we just get, if anything, I've helped ID it, or if I'm following someone, you can do that. It's not really the point, but you can do it. It'll all pop up just on your homepage here. And what I wanted to kind of comment on before I stop sharing this is the City Nature Challenge. It happens every year in April, and it's Canada-wide. It's really fun. There are little bio blitzes in each city where everyone just posts to INAT, and then we all go back later and kind of figure out if anything's new that we haven't seen before in the city or in the province. And it's super good because instead of just having an expert in a really small area, you have tons of people who don't really know a lot, but they're contributing data, and you cover way more space, way more time. It's really great for data collection. So if we just click on that, it's called City Nature Challenge. There's a bunch. If you're in Calgary, there's a Calgary one. If you're in Edmonton, there's an Edmonton one. I think there might be a Red Deer one this year. But you can see tons of people are participating. Tons and tons. And there's one every year. So you can see last year's, you can see 2 years ago, and you can kind of make comparisons on that, which is really useful if you're trying to see like, when does a butterfly arrive in the spring or anything like that. Okay, so does anyone have any questions while I have INAT open? And I can kind of run through anything that is confusing or I didn't touch on.
I have a question.
Yeah, of course.
So I've been using INAT a little bit,
Mhm.
and I've made some accidental, fat finger mistakes. I noticed that you can't even remove those.
You
Like,
mean
is there
like if you
is there
made a wrong ID or something like that?
a wrong accidental ID?
Yeah, so I know it doesn't really matter what we pick. Let's pick this one. So yeah, if I want to change my mind on this, it still logs my old ID. And that's because INAT wants that trail, that paper trail. You want to know what someone has already suggested so that you know if it's the same or if you're changing it or how far out of bounds you really are with your next suggestion. If you're really mad at it and you're like, no, that was an entirely dumb mistake, I want to erase it, you can go up to this edit tab and you have to do it on the web. I don't think you can do it on the app. So it's you might be able to actually. I don't have it open right now, but you could. And you can change your notes, you can add to it. And if you want to change your ID, so let's just change it here to just to genus level. So when we save it, you're right. Okay, so we have to go through here, delete. There we go. The dreaded delete. So now my paper trail is gone. But yeah, so that's only in the kind of rare, when you make some kind of really silly mistake, you can delete it. But even so, like someone else can always come correct it later. It's not really a big deal.
Okay, I can see that that's not available to me on the handheld, on the iPhone.
Yeah, the phone is really easy to use for upload, but I find it is missing some of those kind of editing features. I guess there's trade-offs with the app, of course. Yeah, if one more thing I might mention is that there's these projects that I know some people are curious about. Like, what is a project? What's the point of a project? And these are all just kind of ways to collect observations if you don't have something specific in mind. So if you want everything in a geographical area, it's really easy to kind of wrangle those all together with a project. So like here, this Manitoba nature, it'll just be everything in Manitoba. Do you need a project for it? Not really. But do people like it? Yes, they do. The one that I like is Alberta mushrooms, mosses, lichens, because it's kind of three groups that you wouldn't be able to get in one search because they're phylogenetically quite different. But you get them all in one group in this project, and everything goes in it automatically. So you don't have to manually add it. It's just if it meets those descriptions, if it's in Alberta and it's a mushroom, moss, or lichen, then it pops up. I don't know why some of these are loading and some of them are not right now. Certain pages working, certain pages not working. Anyways, it's a project, you can search it up. If you want to join it, you can join it. But even if you don't, all of your observations that match the criteria are going in it anyways. So the people who do use it, generally, they look at every observation, whether it's research grade or not, and they can kind of pick out what they want or what's useful to their science project based on the geographical loop there. I'm going to stop sharing that since it stopped working anyways. I think it's stressed that I'm making it do Zoom as well. So any other questions from anyone? Anything? Toss them at me.
So the other problem is I can understand the web-based iNaturalist much better than I can understand the app. What I understand of the app is how to add my observation and how to make a guess at what the specimen is.
Yeah.
I can't find all of the things that you're showing me on my app.
Yeah, and the app I find is really good for uploading because it's what you have with you when you're in the field. You can whip out your phone, you can upload a picture, and it's really fast and easy. Even for me, if I'm looking for something in particular, the map function is just so constrained on the small screen. I almost always go to the computer to do that kind of thing. So yeah, like I said, I think the app has its flaws as well as its perks. I personally only use it for uploading, and then I go to the computer for any kind of search or data pulls that I want to do.
Okay, that definitely makes sense to me now. I was feeling so insecure.
No, like I don't find the app is really good for uploading, and for the other things, it's trying to do too much on a little tiny screen.
Thank you.
Right. So I was kind of thinking that if no one else had questions, I don't want this to just be me yapping at you. So what would really be fun, I think, is if we were kind of working together as today is very simple. It's just kind of learning to do it. So we could, any kind of questions, or if you're trying to upload, feel free to ask. But in the future, these kind of Identi Fridays that people host, I was thinking we could do on our Wednesday AMS night, where we all come together and kind of pick a geographical area, say like Elk Island, and work on IDing as many fungi as we can in that kind of boundary. Because a ton of, like I showed you there, a ton of observations are just kind of lost because they're too far away from, usually it's like a university or something like that that would try to aim to identify those, and those ones are just lost. So today, I mean, today's we can do anything. We want to do uploads, if we want to do IDs, if we want to just kind of go off into the night and watch the hockey game, anything's good. But I was thinking from next time, we would kind of crowdsource a good area that we'd want to think about and pull some IDs together. And I would make it so anyone can share their screen. We can all kind of put IDs up. It would be, I think, pretty fun.
So Hannah, there is a question in the chat from
Yeah.
Linda.
Oh, yeah. So what's the best resolution to take photos? I use my camera on my phone only. It's an older iPhone. It does the job. It doesn't have to be a super awesome photo. What I do have and I really like is just a hand lens. So either one that you hold or one that you clip on to the phone. And for those, like I take a lot of pictures of lichens, so they're often a lot smaller. And I find that those photos are better if I actually want a species level ID. For genus level, usually just your camera phone is fine. If you're doing kind of macro fungi, so bigger mushrooms, your camera phone will be totally fine as long as you do the work to make sure you get a picture of the bottom. And if you can get like I mentioned, any kind of bruising information or spore print is always fun. That's really helpful in getting a good species ID. My hand lens, where did I get it? I think I got it like at the university bookstore when I was taking Toby's lichen class, but like you can order them off Amazon. I'm sure you can get them kind of at any naturey store. I can do some digging on that too. Like I know that lots of people I have them for work. I'm sure they get them from somewhere. I bet I could pull it out for you too. It's really tiny. You just kind of flip it open. It's a little tiny magnifier.
So if you would go back to that Gyromitra you were showing, to me that definitely did not look like a Gyromitra. There's lots of brains around the table here. I bet you people would tell you that it's, I don't know, maybe an elfin saddle. I don't know. Let's take a look at it and see what the experts in the room say.
Yeah, of course. And I think that's what would be fun. Like if we're all arguing over an ID, I think that would just be thrilling. Okay, it looks like my project page finally loaded. It just wanted me to turn the zoom off so it could think about itself. But you can see this is the Alberta mushrooms, mosses, and lichens. I'm a member, one of 47, but like I said, that doesn't matter. Everyone's ID or everyone's photo in the geographical range that meets the criteria gets here. So you can see that there's 50,000 observations from that. And if you want to just view them all, it thinks for a second. But like they're all in Alberta, you can see that that line's strictly cut off by the project. And I didn't make that criteria with my search box. It just came like that because I clicked it through the project. And when I scroll, you'll see it's only mushrooms, mosses, and lichens. Not too many mushrooms right now because not the season, but yeah, so I find this is really fun. Like if you care about local species, what people are finding around you, I always check here because I think it's fun to see what's nearby. But yeah, let's go back to what Karen said. So we can go here, we can search Gyromitra, and we can search Alberta. So same thing, 161 observations of 6 species. Let's just check here. We can see some people left it at the genus level. Some went to this subgenus level, but there's not a lot of difference within this group. And then some people have gone for a really specific species, which, as Karen said, sometimes good, sometimes bad. So I think I clicked this one because it was lonely in the corner. Yeah, again, it's from 2006. Like this poor guy has been neglected for years and years, just sitting here. So this is the picture posted, saddle-shaped false morel. Oh, let's do that too. So you can actually click on the ID that someone suggested. And you can see that these are pictures that other people have made research grade for this. So again, it's not necessarily perfect. It's just what's crowdsourced as the photos for this species. So we can see through here, there's a couple different pictures. They're all kind of brown with little tips. So that's good. And we can also, this is really helpful, check on the map. So sometimes someone suggests a species that just, it's only in Europe. And there's a sister species in North America, and they just kind of mixed up which one is where. So if you check the map and everything is in Europe, that's a big red flag, like abort. You got to find some other answer because it's very unlikely that that's actually the answer. But here we can see that there's other observations of this species in Canada and Alberta, so that's not necessarily a red flag. And then we can just go back to that actual observation. So, I mean, I'm not an expert on Gyromitra. I think it kind of fits the pictures that were there, but because I'm not sure, I'm not going to pick agree. I'm not going to suggest anything else just because personally, I don't know better or worse about this particular species.
Can I just say something? I think maybe you're thinking it's a Helvela. Is that what you mean, rather than a Gyromitra?
I did for a moment, but now after I've compared it with the others, I don't think it is.
I think maybe the name has changed, maybe from Helvela to Gyromitra, but I'm not 100% sure on that.
Yeah, and I think too that they started moving stuff to this pseudo Gyromitra. I saw something about it somewhere. I don't see it there, but there's been a lot of juggling from that kind of phylogenetic world. And that's confusing too. So what's nice about INAT is that if the taxonomy changes, your ID will automatically be updated. So that's kind of interesting. If you see something weird, it's probably just been updated to a new genus or a new kind of classification that way. So if we thought this was a Helvela here, we could always type it in and then just click this view. It doesn't change anything on this person's observation, but it pulls up, like I said, those pictures that other people have kind of crowdsourced to be this genus. So we can see it has kind of the same white stem, but it's kind of like a Swiss cheese stem versus the other one looked more like a solid tube. It's a different color. They do look similar, but I do think the Gyromitra was probably a better fit for it. So I would leave it at that. And that's fine. I mean, if you don't know better or worse and you want to just leave it, that's not a crime. Like, don't feel like you have to push an ID to get something to research grade. It's totally fine for this to sit here since 2006 if no one can confirm or deny. This species probably, I think there's a couple of Gyromitras that are really similar. And if you don't have a microscopic kind of like proof evidence, then you can't really conclusively say one or the other. So that's fine. It can sit there.
Yeah, I think it's Gyromitra infula.
Yeah. So I won't do it, but if you go in and you search Gyromitra, then you can put your name on it and say, yes, I think it is. Because yeah, like I am not a Gyromitra expert. I don't really know. I go out and I say, I can get it to genus level. And that's about it. So yeah, let's go to identify. Let's see what's out there right now. It's always changing. Sometimes people do big mass dumps of pictures and it's really fun because you get a whole bunch at once that are new. If you like birds, there's always birds on here too that need your help. So we can go here. All of the fungi, including lichens in Alberta. And because it's spring and nothing's really growing, it's always a lot of lichens and mosses and stuff, but that's okay. Works for me. Yeah, so like this one here, I can click on it. I know that the basics for Xantho Mendoza phalax is these little mouth-looking things with powder in them. And you can see this person has captured it super well. It's a yellow lichen with these edges that are powdery, and there's kind of like it's called hooded sunburst. So sometimes people think of them as hoods that are holding that powder. So I would say I'm pretty confident in that being Xantho Mendoza phalax. And that's just because I like lichens, so I know that that's going on. And now this is research grade. Because we both agreed at the species level, we were exactly sure it goes. If this person had put it at genus level and then I suggested a species, we would need a third person to kind of confirm that it's that species. So I just kind of want to, where are we at here? Oh, Hannah, how do we find you in INAT? You can if you want to. My username is Hannah DV. And if, like I said, you can follow me if you want. What it'll get you is a list of my observations. So when I post something new, it'll show up right on your homepage. If you want to see how many lichens I see, you go for it. And that's about the only benefit is, so if you were following someone who was studying something in particular or was on a project you were interested in, you could see what they were contributing kind of live time, depending how often you check INAT. Other than that, it's not really like a social media platform in that way. But yeah. Does everybody kind of feel like they could upload something by themselves without any major issues? That's what I really, oh, one person says no. All right, let's do it. we are going to upload something together with no issues. I'm going to find a mushroom on my computer, which of course I have. Let's do, oh yeah. All right. So we're back to iNat. I'm going to go to this top corner that says upload. Drag and drop some photos. I'm going to choose my files. I'm going to pick this. You can see that this is actually my photo now, so it pulls the date, the time, and the location. Let's ask it what it thinks. You don't have to. If you already know what you think, then just ignore the algorithm. It's not smarter than you. And it says it thinks it's this genus. It might be these suggestions. This is fun because now it's pulling anything pink. It says maybe it's this mushroom, maybe it's this slime mold. It's got, wow, it's really reaching here. Raspberry slime mold. That's cool. So if you know your wrinkled peach, this is a wrinkled peach. And what might help someone else confirm my ID is I might put found on elm log. And wrinkled peach loves elm, so that would be a really helpful comment for someone who's coming to my observation. And like, let's say I took a really blurry picture, that comment would probably help them confirm my ID. We can go here. If you found a morel patch and you really want to keep it a secret, but you also want to share your knowledge with the world, you can put this location as obscured. And instead of putting like a pinpoint on the map, it puts a broad circle. So they know that it's somewhere in that region, but they can't just walk up to the GPS point and say, yes, this person's morel patch is here. So some people do that if they're really protective of their edible mushroom patches. If an animal, if you're doing like a grizzly bear or something, sometimes when it's like categorized as endangered, it'll automatically do that. Or someone who's actually a staff member at iNat can change it manually for you if that animal is at risk of being hunted based on your GPS point. But that's not really relevant to a mushroom anyways. So let's say I'm obscuring this as my personal wrinkled peach. If you're doing like a tree in your backyard or something, you can click captive, puts this little C on it. It just means you didn't find it in the wild, which can be helpful if you're looking at, let's say, an invasive plant, but you just have it in your garden. That's a big difference between knowing if it's spreading versus just having it in your garden. You can put these tags on it. You can put whatever you want. You can put like edible. I don't find it's very useful. If I wanted to add it to Alberta fungal biology observations, sure, but I don't because it's not. There's a couple that I pull up. These are projects that you have to purposefully upload to versus ones that are just geographical pulls. So I'm going to ignore those. And then you can also add, like, you want to specifically say this was growing on an elm, you could do that. But again, it's just bonus information for someone else or for you later. You don't have to do any of this. So that's my picture. Submit one observation. Wow, it's really thinking. There we go. Boom, done. So these are in reverse order of my dates. So there we go. We can see that I just uploaded this wrinkled peach as of April 25th, 2024, which is today. And I'll go delete that later because it's a duplicate. But for now, that's all good.
Can I also just add that you can add numerous photos to an observation?
Yes, of course. Yeah.
For mushrooms, yeah, which is handy for mushrooms because sometimes you want to see the top and the underside and sort of where it's growing. So yeah, it's not just one photo.
Yeah, that's totally right. And like you said, that can be really helpful, especially like, let's say you have a picture of it in the ground and then you pick it and you have more details. Those are all good. And like, I could choose as many of these as I wanted. I could pick this and I don't know, this, none of them match, but I can upload all of them. And here it's going to make them all separate. So you could do a bulk upload from your camera, or you can go in and add more to each specific one, which is usually easier to do on the app, but that's just my personal preference. So there are lots of ways to make this easier for you. If you have your camera, I would recommend the bulk edit online, which you probably plug in your card to your computer anyways, but it's a lot easier to do from here where you can just click them all and they all come up as their own observation. So you can see in the corner, it would say submit 3 observations. So these would all be separate, or you can add multiple photos to a single observation. Either way, like whatever works for your situation, you can make it work. Okay. How do you create a project? Oh, let's start here with how far back should we go? Can you upload a video? Yes. So let's go to identify just to see what's up there. Oh, I got a mail. Someone following me? It's Rick Watts at Elk Island National Park. Perfect. I'm gonna ignore it for now. Anyway, so here with all of Canada, let's see if I can pull it up. I'm gonna go with birds. There's tons of bird people who love INAT. I'm not sure if, oh, here it is. So you can actually upload a video or you can upload only a sound. So if you saw something that, I don't know, it's not really a mushroom thing, but if you saw a bird and you were just recording it and you didn't know what it was, you could actually upload only the sound and you would get that ID'd potentially by someone if they cared to listen to it. So that's a really cool one for bird people. And you can also upload a video, I believe. I've never tried it. Again, like I usually just take a picture of the top and then I flip it over and a picture of the bottom and that's that. But I think you can do it. I wish I could ID birds. They're all so cute, but I have no bird knowledge. Yeah. And then another thing you could do if you're feeling really motivated to kind of help people who are really lost is you can turn on this. You can see these categories. It's like birds, amphibians, just general ones. There's a mushroom. And then there's this sad question mark of unknowns. So if you upload only a picture and you don't assign any kind of ID to it, it gets thrown into this sad unknown category. I had it on birds, so there's obviously no oh, here's a great one right here. Let's do it together. So someone said this was just unknown, no idea. Oh, look, okay, so it's Elaine's first observation. So we can take pity on Elaine because she's never done this before. So she uploaded it as just a question mark, no idea. So if I was to go in and say, I'm really good at identifying fungi, so I'm going to search fungi and do all my helpful IDs through that, I would never see this photo because it's just unknown. She put a placeholder that says Morel along trail, mixed spruce, aspen forest. Helpful to me, but not to anyone who's Googling Morel or fungi or anything like that. So we can see what the algorithm suggests. Thinks it's a morel. It has some species suggestions. Is anyone good at morel species? They want to tell me what it is. Myself, I would pick just genus because like I'm not good at the to species level all the time. Maybe someone else knows. I'll go up to this picture. That's all you get. All right. No call outs. It's going to be just true morels. And that's totally fine. Like putting it at the genus level, no harm, no foul. You've gotten it 90% of the way there. So that's honestly pretty great. So then I click done. It loads for a second. True morels. Hurrah. And so now if I search fungi or morels or anything like that, it's going to pop up in my search. And it was right near Nordic. And it was in June. Okay, so she submitted it today, but it was observed June 16th of 2022. So that's helpful too. That gives you a timeline for when you expect it to be there next year. Very handy. I can manually put this in a project if I want to, but again, you only need to do that if it's one of those projects that has manual uploads. And most of the good ones aren't like that anyways.
Well, we are nearing the 8 o'clock hour. I'm sure that you have the ability of catching all our email addresses so that if we want to join you